Hey, T-Sauce, Andrew here. Today we will look at a new drug that just dropped in the field of Alzheimer's disease research as of a few months ago. It's had a lot of people excited and hopeful, and some not so much. A company by the name of Neuromune has found a way to produce several antibodies that target things called plaques in the brain that build up and are highly associated with the characteristic cognitive decline in Alzheimer's patients. First, let's look at a common protein in the brain called amyloid beta, which is formed when the amyloid precursor protein, or APP, is cut in a very specific way. Imagine taking scissors and cutting a star out of a piece of paper. That paper is APP and the star is the amyloid beta. While these amyloid beta molecules are natural in the human brain, overproduction leads to plaque formation, as amyloid betas clump together and form an aggregate, hindering our cells' ability to communicate due to excessive inflammation as a bodily response. The increased production of amyloid beta are often the result of mutations in our genome, such as chromosome 21. Neuromune initiated this process by taking the genetic information from individuals that have not yet developed Alzheimer's, with the supposition that somehow these individuals have some sort of inherent resistance to the disease. These antibodies specifically target the aggregated amyloid betas because, as we mentioned earlier, non-aggregated amyloid betas also exist in our brain naturally. It is possible to only target these structures because antibodies are highly specific to one single part of the protein. So researchers are able to target one part of the amyloid beta that isn't usually visible unless they are clumped up. This is where other antibody treatments such as the ones by Pfizer had failed because it binds to the regular non-conglomerated form. Once the plaques have been tagged with several antibodies, the immune system comes in to deal with the mess. Microglia, as part of the brain's immune system, comes in to digest the plaque complexes, thereby reducing the amount of amyloid beta in the brain and theoretically improving cognitive function. An interesting side effect of this drug is that tau tangles, which are another contributor to Alzheimer's, were also found to decrease. In order to keep our cells semi-rigid and not completely collapse at any point in time, there exists a sort of scaffolding, which also helps to deliver nutrients and such, called the cytoskeleton. In neurons, tau proteins help keep the cytoskeleton in place. However, in Alzheimer's patients, tau proteins are modified in such a way that causes them to break away from the scaffold that they are part of and clump together in what is called a tangle, much like the amyloid beta, except the tangles here are inside the cell instead of outside. This causes the cytoskeleton of the cell to fall apart and the cell to eventually die off. So, while tangles are a major concern for Alzheimer's, the initial prospect is that this drug might be able to help. This newly designed drug, called Aducanumab, will be improved upon to target these structures and plaques with equal efficacy. In the typical sequence of clinical trials, respectively phase 1, 2, and 3 tests its baseline safety, actual effectiveness, and for appropriate dosages to be administered. To support their theories, Biogen, who is licensed by Neuromune to use Aducanumab, had actually performed 4 phase 1 trials to test effectiveness and phase 2 trials for safety. These were nicknamed Prime and Evolve for phase 1 and 2. In all of these trials, Biogen used individuals with confirmed plaques scanning for known Alzheimer's mutations and using PET analysis, or PET analysis, as well as a confirmed Alzheimer's diagnosis as deemed through the mini mental state examination and the clinical dimension rating sum of boxes. Participants took varying amount of the drug or placebo and then continued reception of the drug every 4 weeks for up to a year. According to the phase 1 study, Aducanumab has dose-dependent reduction of plaques, with plaques reducing in several areas of the brain. In addition, the test for cognitive function, the 2 qualitative examinations, also showed a decline in score correlated with the dosage, which indicated improvement in cognition. Armed with these promising phase 1 and 2 studies, Biogen initiated 2 phase 3 studies. They called that Emerge and Engage. While the Emerge study found similar if not better results to the phase 1 studies, slowing cognitive decline by 25% and functional decline by 40%, the Engage study showed no differences between experimental and control groups. The key to the Engage study failing was an amendment made halfway through the study, which the treatment dosage was halved in groups susceptible to unnatural damage from the antibodies in the brain. This amendment had to be approved by the patients, which took 18 months to implement, skewing results significantly. However, in those groups where the dosage remains the same or even more, The findings were consistent with eMERGE and other successful trials. Decline in the qualitative study scores were slowed in both eMERGE and ENGAGE. The FDA has granted Aducanumab a priority review, which is an expedition process to approve or deny a drug that is currently in trial. However, while the FDA itself is complimentary of Aducanumab, the Independent Advisory Council to the FDA is not so keen, since one of their two most recent phase 3 studies have shown no difference in cognitive decline as compared to the placebo, or no effect. The council voted 8 out of 11 to reject this drug. 2 had said they were uncertain if there were any effects, and 1 was positive about the drug. In addition, the critique of this council has concerns about the damage to the brain related to antibodies. While some of the council might be looking down at the drug, various members of the community are hopeful, looking for treatment that is novel and has promise. For decades, Alzheimer's treatment has been stagnant, with rejection after rejection, but this is the first antibody drug to be expedited for approval. Geldmacher, a scientist at Biogen, surmises outlook on this drug as opening the door to what future success might look like. Even if the drug fails, he says that it has a proof of concept, that these concepts are able to induce positive effects in Alzheimer's. In light of the data and results, one cannot help but feel a little bit optimistic. The mechanisms at play are proven concepts in immunology and additionally have improved on the faults of the previous antibody therapies. Biogen was able to prove, with statistical analysis, that antibody therapy reduces the progression of Alzheimer's, both in terms of plaque and tangle reduction and decreasing qualitative dementia scores. The ENGAGE trial that failed had a plausible reason behind it, and the subgroup that was unchanged still reproduced expected results. Thus, the outlook is bright on this drug. Maybe Aducanumab can be the pioneer for new types of treatment, not just Alzheimer's, but also for many other neurodegenerative disorders.